Hello everybody and welcome to the orientation video for the 2026 CompuCell3D Multiscale Virtual Tissue Modeling Workshop and Hackathon. My name is Hayden Fennell. I am the organizer and coordinator for this year's summer workshop, and I will be walking us through the orientation materials today. Just a few notes up front. This slide deck is available at the following link that will be in the video description below, so please go down to the description to find the link to the student materials folder for this year, which will contain this slide deck as well as other materials that we will discuss today. We do have a Slack channel set up for the workshop this year, so please go ahead and join the workshop Slack channel whenever you get a chance. We'll talk a little bit later in the orientation about how we use that channel during the workshop. One important note is that workshop sessions will all be live streamed, recorded, and distributed on YouTube. Just so that everybody is aware, the sessions will be live streamed to YouTube during the session and then edited lightly and made available on YouTube in perpetuity. So if you do have any issues with being recorded or having your likeness or voice out on the internet, please do let us know in advance. One other note is to please do download and install CompuCell3D to your personal computer in advance of the workshop. We'll talk in actually a second part 2 video about how to do this, but you will need to have CompuCell3D installed on your local machine before the workshop begins. We will offer a couple of installation troubleshooting sessions in July of 2026. If you're having trouble getting it installed on your particular system, we will be available for help. You can always also email us at any point, and we can try to set up a one-on-one meeting if you're really having trouble with that. But we do not currently have a remote operable version of CompuCell available for use, so you will need to make sure that it is running on your personal machine before the workshop starts this year. We will have very limited time to troubleshoot problematic installations during the actual workshop. And lastly, we'd also like to thank our funding sources through the NSF, as well as our previous funding sources through the NIH and some other NSF grants. So today's learning goals, we're going to discuss kind of 4 main things. First of all, what is virtual tissue modeling? Why do we need it? What is CompuCell3D and what is it capable of? What has it been used for in the past? What are you going to have to know to work with CompuCell3D? Specifically here, we're going to identify some things that you might want to read up on a little bit before the course starts if you want or need. We will teach you everything you need to know to use CompuCell during the course, but if you'd like to really get the most out of it, there are a few areas where we feel like you might want to do a teeny bit of reading over the next couple of months as you prepare for the workshop, and we'll provide some suggested resources for that. Again, I want to make clear you're not required to do any reading in advance, but it will make your time during the workshop even more useful and meaningful if you've made sure to kind of fill in some of the areas where maybe you're not as familiar, and we'll talk more about that in the second half of the orientation. We'll also talk about where to find resources for the workshop and for filling in background material. So a little bit about virtual tissues. This may be the first time some of you have heard this particular term, those of you coming from a more traditional biology background or other backgrounds besides computational biology in particular. So virtual tissues are essentially mechanistic computer simulations of cell and tissue organization. These also look at emergent dynamics that are characteristic of biological function. Distance scales can range from sub-nanometer to meters, so all the way from the microscopic to basically what we can see with our own eyes. And time scales range from matters of seconds to matters of decades. Where CompuCell is kind of situated in this broad range of possibilities for virtual tissue simulations is here in this dotted blue line on our graph here. So you can see here we have representations of increasing or decreasing scale from whole animal simulations to organ level, tissue level, the multicellular level, intercellular, which is more signaling between cells, within cell behaviors, and then subcellular scales. CompuCell is situated kind of at that organ to tissue to multicellular level simulation. So most of what we'll be looking at with CompuCell is looking at groups of cells interacting and how they come together to form tissues. We will be talking a little bit about intercellular signaling with network modeling, which can be done in CompuCell as well. But for the most part, most of the examples we'll be looking at will be at that kind of more tissue level simulation. So why use virtual tissues? There's a number of reasons why we use virtual tissues. We're just going to highlight a few of them here. One of the big ones is that they function as a variable magnification virtual microscope that allows us to study the processes at variable spatial and temporal resolution, so we can actually look at a phenomenon at different scales using kind of the same simulation architecture in a way that is more difficult to do experimentally. It can infer hard to observe parameters and test mechanistic hypotheses. So once the simulation has been sufficiently validated, we can use it to observe elements of a phenomenon that maybe are difficult or sometimes even impossible to observe experimentally. Another big one is that it reduces the need for animal experiments. If we can simulate something in silico that does not require the use of live animal experiments, that is always a benefit. Again, allows for the testing of situations that are impractical to test in experiments. It allows us to visualize and communicate systems level outcomes. One of the nice things about virtual tissue simulations is that it's very easy to generate intuitive and interpretable visuals to pair with our output data. And lastly, the big point, or one of the other big points, is the idea that by building models, we can codify and test our biological understanding. Often the process of putting models together will reveal critical gaps in understanding and experimental data. The virtual tissues are not just useful in and of themselves as knowledge generation tools, they're also useful in identifying current gaps in the literature, identifying what hasn't been done yet, and potentially generating new questions from our simulations. Specifically, what will we learn to model during this workshop on CompuCell? We'll talk a little bit about development, so how does a fertilized egg organize into an adult? Homeostasis, how does an organism maintain itself? So once it has developed, what are the processes behind the cells maintaining themselves? Basically, what does it require to remain healthy and behave as expected? Talk a little bit about developmental diseases, how issues of homeostasis during development lead to different pathologies. Infectious diseases, how pathogens and hosts interact. We'll look a little bit at toxicology, how certain compounds cause damage, why they cause that damage, what determines susceptibility to that damage, what concentrations of chemical compounds are needed to reach critical thresholds of damage, questions along these lines. And we'll look at the big question of medicine and bioengineering, which is how can we control, repair, or create new forms of these processes. So by using our virtual tissue simulations to kind of understand the underlying mechanisms behind these different disease pathways and toxicological information, we can tune our simulation to kind of show us how we might approach reducing the harms or effects of these and developing treatments that will counteract them. All right, so next we're going to look at a few sample virtual tissue applications in CompuCell3D, just to kind of give you an idea of the variety of different things that you can model using the software. We have various papers here that have been run by different people and different groups. On the top left, we have a model of polycystic kidney disease, which is looking at the roles of cell adhesion and proliferation in the onset of kidney cystic disease and how polyps form on the renal tubes in the kidney. In the upper right here, we have a video of tumor interaction with blood vessels as it grows within a vasculature network. You can see the vasculature there in red and the tumor growing in green, kind of disrupting the vasculature. Vasculature, you'll see, reconnects at different points, and it's also modeling how the tumor is sapping nutrients from the vascular network as it grows. In the lower left here, we have an example of diabetic retinopathy therapy optimization modeled in CompuCell3D. And this is basically looking at how different patterns of targeting laser treatment on the retina can produce different outcomes in the treatment of the progression of the disease, basically used to optimize the treatment to help slow the progression as much as possible. And then in the lower right here, we have a video of a simulation of retinal neovascularization. This is a condition where the blood vessels behind the retina begin to grow through or up into the retina. You can see in the cubic volume here, we have a 3D simulation of the blood vessels growing through the vasculature there. And then on the left, we have a 2D view, and you'll see that as the blood vessels kind of progress further and further across the retina, eventually we will have detachment of the retina, which we should be seeing here in just a moment. Yep, you'll see there it lifted off of the substrate in the simulation that the retina was attached to. So these are a variety of different types of simulations, all being run using the same software platform of CompuCell3D. Again, just to give you a feeling of the variety of different things that can be done using the software. There's also a number of toxicology studies that have been performed using CompuCell3D. I won't go into too much detail here about these, but you can see it's been used in how to predict different methods of blood vessel or disruption of blood vessels during development. It's been used to model genital tubercle development. It's also been used to model secondary palate fusion and disruption. So while the palate is fusing at the top of the mouth during development, disruption of that process results in a condition known as cleft palate. Like with all these previous examples, understanding the mechanism of how the tissues actually form during palate fusion and how those can be, or how those are disrupted, can help us target treatment early on to avoid these conditions. So just a couple more examples here. CompuCell has also been used to model viral infection and immune response in epithelial tissue. So this was a study done actually during the early days of the COVID pandemic, where you can see how the virus begins to spread through epithelial tissue and the immune cell response to that. This is actually runnable online at nanohub.org at the link listed on the slide. Again, this will also be in the video description if you'd like to go check that out on NanoHub. But this is just to kind of demonstrate that we can also use CompuCell to do infection and immune response modeling. So a lot of different things we can do with CompuCell using the same underlying algorithm, which we will talk more about on day one of the actual workshop. So by the end of this course, you will be able to look at a biological problem and ask quantitative questions that can be answered with simulations. It's the first step, is being able to break a problem down into components that can actually be meaningfully answered with a computer simulation. You'll also be able to analyze the components of a multicellular system and their interactions and translate them into executable CompuCell3D code. And then you will be able to build and modify CompuCell3D simulations using our XML language, CC3D ML, and Python, using CompuCell Wizard, our editor called Twedit++, and you'll also learn how to display and analyze simulation data to derive biologically meaningful results. So during this workshop, you will get a crash course basically on all stages of the modeling process, going from conceptualizing a problem and turning it into a model that can be run in a computer, and then actually writing that computer code and analyzing the output. Okay, so next we'll talk a little bit about course logistics and what the workshop will actually look like in practice. So we'll be sending out Zoom links. The workshop will be run through Zoom. We'll have the links set up and sent out about 2 weeks before the workshop actually begins. Those links are still to be determined, but we will send them out multiple times in advance of the workshop. So please do make sure that CompuCell3D.iu@gmail.com and hfennell with one L at iu.edu are added to your allow list. And keep an eye on your inbox. Kind of mid-July, we'll start sending out announcements for where to go on day one of the workshop. So please do make sure that you have a local version of Zoom installed on your computer, and that it has been updated before the workshop starts, and then it's usually a good idea to check for updates when you log in each day. Some of the features in Zoom don't work if the host and attendee clients are out of sync version-wise. The most common thing that gets turned off is polls, which we use a lot to help manage the pace of our sessions throughout the workshop. So please do make sure that you have a local installation of Zoom and that it is up to date at the start of each day. Unfortunately, the browser version of Zoom will work in terms of logging in to watch the video, but some of the other features that we use, like breakout rooms, polls, some chat features, things like that, are not available in the browser version. So we do recommend heavily that everybody has a local installation. So some of the people, our main teaching team this year will be Professor James A. Glazier at IUB, Professor Gilberto L. Thomas from Brazil, Mr. Pedro Sensi Dal Castel, also at IUB, Professor Julio Belmonte from North Carolina State University. Dr. Lorenzo Veschini will be here to help teach a couple sessions and also to help out as a TA in the chat for us. He's also with Indiana University Bloomington, IUB. We'll have Dr. Priyam Adhyapak from Duke University, Dr. TJ Sago from University of Florida, Dr. Michael Getz from IUB, and of course myself, also from IUB. Emails are here on this slide. If you need to get in touch with any of the instructors directly, you are welcome to email them. If you're not sure who to get in touch with, you can always email me, and I will forward you to the most appropriate person if I can't answer the question myself. But don't hesitate to reach out after the sessions to the instructors directly if you have specific questions for those instructors. Some Zoom logistics. In general, for the sessions, screen sharing will be disabled and microphones will be muted by default when you join, except in breakout rooms and for special sessions. If we go to breakout rooms for an activity, obviously you'll be able to talk to people in your breakout room, but for the main session, we will have microphones muted by default. You can put any questions or needs in the Zoom chat while we're going through each session. We'll have a couple of people monitoring the chat during each session, and if there are particularly relevant questions in the chat that we feel need to be forwarded to the instructor in real time, we'll break in and ask them for you. But we'll make sure that each session has several breaks where the instructor can stop and check the chat for questions to make sure all of those get answered. If you need personalized attention for any reason, you can let us know. But sometimes we'll have people available to move to a breakout room and help you. For example, if you're having an issue where you're struggling with your software or something like that. Generally, we try to keep assistance in the breakout rooms to a minimum if possible. One thing we found is that when you leave to go to a breakout room for 15 minutes to get help with a specific issue, when you come back, you've now missed 15 minutes of the main session. So we do try to balance that concern. If you are having a serious issue with your software, we can try to help during the breaks or at the end of the day. But in general, we recommend that people ask questions in the chat, and the moderator and TAs that are available for each session will help answer those. Zoom does have buttons for go faster and go slower on the participant screen. Please do feel free to use those if you feel like we're moving too quickly or too slowly. We will have polls during exercises for level of completion of an activity and if you need more time. Don't worry if some exercises are taking a long time to complete. Some of them are kind of intentionally designed to have multiple pieces that everybody may or may not get through. So don't worry if you can't finish every exercise completely. We'll make sure that we have people available for assistance if you would like to complete those, offline during the workshop period. All exercises will be made available in Google Doc format and as PDFs, so you can continue working on them offline. We'll have those in the student materials folder each day for you. Videos and PowerPoints will be made available for you to review if you miss any materials. So all of the course slides will be in the student materials folder, and each video from each session will be available usually about an hour after that session has streamed. It does take YouTube a little bit of time to finalize the processing on the video, but usually within an hour or so those videos are up if you would like to revisit anything. All right, a little bit about the daily schedule for each day. Each day will follow the same general template. All times here are in Eastern Daylight Time, which is UTC -4. Please do keep in mind the Daylight Savings Time issue. Again, we're in Eastern Daylight Time, not Eastern Standard Time during the workshop. So from 9:45 a.m. to 10 a.m., we'll have a brief discussion, troubleshooting, and questions with instructors session. So if you have any quick questions or any small issues that you'd like some help resolving, you can show up a little bit early at 9:45, and we're happy to talk with you then. From 10 a.m. to 11:30 a.m., we'll have our first module of the day. These are usually 30 to 45 minutes of lecture with 45 to 60 minutes of hands-on exercises kind of interspersed throughout the session. Then from 11:30 to 12 p.m. or 11:30 a.m. to 12 p.m. we'll have what we call our extra session. This time is used for addressing shorter topics throughout the workshop and also allowing some time for hackathon teaming. The full schedule of extra sessions will be available in advance of the workshop a few weeks before we get started. From 12 p.m. to 1 p.m. we'll have our lunch break. From 1 p.m. to 2:30 p.m. we'll have our second main module of the day. From 2:30 to 3 we'll have another short 30-minute break in the afternoon. From 3 p.m. to 4:30 p.m. we'll have our third module. And then from 4:30 to 5 p.m. we'll have discussion, troubleshooting, and questions with the instructors. So all of the instructors for the day will be available in that 30-minute Q&A session. Usually we can go a little bit longer if we need to, or if you need to ask specific questions of specific instructors, we'll have breakout rooms available for that. But if you have any questions at the end of the day, we reserve that, at least that 30-minute block, for just addressing questions and having some time to talk with the instructors. Okay, so next topic of discussion is a little bit about background knowledge and reading. There are a few things that you might want to look into a little bit before the start of the workshop, and we'll go over what those things are now. So before we get started here, though, I do want to just emphasize that we don't expect everybody to come in as a content expert on any of these topics. They are intended to be things that, as we walk through them, if the topics look totally unfamiliar to you, it may be worth taking a little bit of time to gain just sort of like a Wikipedia level understanding of some of these topics. Again, it's not required. We will teach you everything you need to know for the workshop, but having some familiarity with the concepts before you go into the workshop can really help you get the most out of things if you have time to do any preparation. So there are 5 kind of primary background areas that we think are the most useful when thinking about computational modeling. Those are the principles of modeling, Python programming. We will use Python a good bit during the workshop because CompuCell simulations are scripted in Python. Obviously for computational biology, you'll need to know a little bit about biology. You'll also need to know a little bit about biophysics. This is how we translate from our biological systems into mathematics that are then run in a simulation. And then, of course, following that, you need to know a little bit about mathematics. So again, you don't need to be a content expert in all or even any of these topics. This is just to give you a feel for what kind of reading you might want to do before the workshop if you have time, and we'll point to some resources here at the end of this section for you to do just that if you'd like. So first topic is background principles of modeling. This is kind of covering why we do modeling and how to approach the modeling process. So some things maybe to look into here are defining questions that can be answered by models. Not all questions are answerable by computer simulations. Selecting meaningful variables and parameterization is also an important process. Being able to decide what components and mechanisms are important for your simulation is kind of a big one. All simulations are to some level simplified abstractions of the real world, and we have to decide during the modeling process what components of that large and very complex real-world system are necessary to include in our simulations to answer our questions at the end of the day. Understanding the limitations of your models, so what they can and can't do, as well as what parameters over which your simulation holds. Often your simulation will be good for a range of different starting parameters, but once you go below or above those kind of accepted parameters, the model tends to sort of fall apart. So understanding what your model can and can't do and what it is and isn't intended to do are very important first steps as well. So you'll also need to know a little bit about Python programming. You won't need to know a whole lot of Python for CompuCell, but CompuCell does run its simulations using some Python programming. So some of the things you'll need to understand, we'll just walk through the list here, is basic syntax and indentation. Here we have the example of the double equals. Understanding the difference between a double equals and a single equals is an important example of understanding Python programming syntax. If you don't know what the difference is, then it's probably worth spending a little bit of time brushing up before the workshop. You'll need to know things like loops and Boolean logic and conditionals. You'll need to know about object types, lists of information, and dictionaries. Lists are taught pretty commonly in most Python tutorials out there. Dictionaries are covered a little bit less often, but CompuCell does use dictionaries to keep track of a number of things during its simulation, so it is important to know about dictionaries so you can better understand how your simulation information is being stored during the simulation run. It's important to know things like what is a class, scoping and inheritance. We won't do a whole lot with classes and inheritance in CompuCell3D, at least not during our workshop, but it is useful to know what it is, because some of the built-in structure of CompuCell makes use of classes that inherit values from each other. You'll see this works with the self.dot class. Self.dot is used a lot in CompuCell. So if you don't know what the self.dot class is, we recommend looking that up. And it is useful to know about attributes and methods as well, so writing your own functions in Python. Again, you won't need to know a lot of Python. You do not need to be a Python programming expert to interface with CompuCell, and we will teach you everything that is relevant to know during the workshop. But we do have a couple of resources at the end of these slides for going over some of the basics, and we also will be offering a relatively brief video series on preparing for Python in CompuCell before the workshop. So a little bit about background biology. We're not going to go into all of these in super great detail, but just some questions. Again, these are up here more just to assess your own familiarity with the kind of the key underlying biology. Some important questions, or some things that are useful to know, since we'll be using CompuCell to model cells, virtual tissues. What are cells and what do they do? Kind of fundamental question there, but how do cells generate forces? How are tissues constructed? What is cell polarity? What's the extracellular matrix? How is it different from the cell? How is it generated and what does it do? How do cells communicate? And then kind of a bigger one here at the end, or at least a more general one, is how do biologists represent information? We have on the right here an example of a cell walking cartoon diagram. Biologists will often use these cartoon diagrams to illustrate ideas that are hard to express just through language. But you will notice that these diagrams often have standardized components in them to kind of cut down on the amount of explanation required in the diagram itself. So understanding a little bit about these cartoon diagrams can often be helpful, particularly if you're looking through papers to, for example, prepare for the hackathon or looking to put together a project of your own. Having seen some of these things before and knowing what the different pieces mean can be very helpful. So next, a little bit about the background in biophysics that you'll need. We have a few points here. The only one that we'll really talk about here at all is this idea of what is diffusion. It's also important to understand what a constraint is, how constraints combine and interact. We'll cover how constraints interact in CompuCell during the workshop. But understanding diffusion is probably the big one on this slide. We will be using diffusion a good bit in CompuCell. So it's not, again, you don't need to understand it in super in-depth, but once we start talking about chemical fields in CompuCell, we will be talking about diffusion a good bit. So if you've never heard of diffusion before, at least from a biophysical perspective, it's probably worth reading up on the Wikipedia page again about that. And finally, for some relevant topics in mathematics that might be useful to look into ahead of the workshop. Again, we have a few points here that might be useful depending on your particular project and simulation ideas. But the big one that is the most useful to know is a little bit about first order ordinary differential equations and rate laws. We won't be talking super in-depth about the mathematical components of biological modeling during our CompuCell workshop. We will bring them up as necessary to kind of explain how a model is set up. But having some familiarity with ordinary differential equations and rate laws and how they're represented in biological settings can be very helpful, particularly if you're planning on kind of writing your own equation set for a simulation that you would like to develop for the hackathon or after the workshop. And as I mentioned earlier, we do have some resources for places to fill in this background that we think are good starting points. For principles of modeling, we have a couple of links here. One paper that we like to reference people to is this 10 simple rules for building your first mathematical model. Again, we have the link here. And then we have some resources of our own here on the base modeling workflow. For Python, the official Python tutorials are pretty useful, but they do tend to spin into sort of advanced Python user territory relatively quickly. So one resource that we like to point people to in addition is the Python Village set of exercises from Rosalind. This is a series of, I believe, 6 sort of multi-part exercises that will show you the basics and then have you do activities interactively through them. And we've found that if you can complete the first 5 exercises of that 6 exercise set, you're in perfectly good shape for the workshop. And then we have a book recommendation here that we think is useful for beginners. For network modeling, we have a couple of resources. We'll be talking about network modeling on day 5 of week 1 of the workshop. But if your project is going to be very network modeling heavy, we have a couple of places here where you can learn a little bit more about it. For math and biology, we have a couple of good textbooks. Wikipedia has good articles on these concepts as well. And with these textbooks, you'll find that the more recent versions are obviously more expensive, but the base mathematics and the base biology involved in most of this stuff hasn't changed in a very long time. So ordering older editions is often an easy and much cheaper way to gain access to this information. And same thing with our physics textbook recommendations here. All right, so next we're going to go over some quick details about the hackathon. The hackathon will take place on August 7th, 8th, and 9th, following the advanced week of the workshop. This is a chance to practice the skills that you've been developing over the course of the workshop by implementing a simulation of your own. Now, you do not have to have a project idea to participate in the hackathon. We are happy to help people join groups. So as I mentioned a minute ago, the hackathon will take place from Friday, August 7th to Sunday, August 9th. We will have at least one hackathon teaming meeting in advance of the workshop where people can come and give lightning talks of 3 to 5 minutes each to pitch their project idea for the hackathon. We have a template available in the hackathon folder on the student materials drive that you can use to help prepare a short presentation about the problem you want to model. You do not have to have a project idea to attend these project pitch meetings, and we actually would encourage everybody who can make it to come to those so you can see what kinds of projects are currently being pitched. If you're looking to join a team, coming to these project pitch meetings is a good way to kind of get started thinking about what kind of project you might want to work with. We will help organize the teams before the start of the hackathon. Often these teams tend to be sort of self-assembling, but we are more than happy to help people find a group if you would like to participate and aren't sure exactly what you want to do or where you might be most helpful. For a little bit of structure on the actual hackathon, we'll have brief check-ins in the morning and at noon each day with the whole group. This is a chance for project groups to kind of give us an unofficial or informal report of how things are going, what kind of specific help they might need. But other than those check-ins, you'll spend most of the day in your group's breakout rooms just working on your model, and we'll have instructors circulating around to the different groups to help people answer questions and debug and things like that. Each day we'll have an evening report so that we can have feedback for you the next morning. Or on Friday and Saturday evening we'll have an evening report, again a brief informal report. And then on Sunday evening we'll have a very short oral presentation from each group where you can talk briefly about the biological background of your model, how far you got into the model during the hackathon, and what you plan to do with it after the hackathon, if anything. And on that note, we will be available after the hackathon to continue to support individuals and teams who would like to continue to work on their projects. We've had a number of publications that have come from hackathon projects initially, that groups that we've worked with over the years. So if you're interested in turning your project into a publication, or if it's going to be part of your thesis maybe, or if you would just like to continue to work on this out of your own interest, we are happy to continue working with you after the hackathon. Okay, so we are almost finished here. Just to talk a little bit about where to find resources for the workshop itself. Not necessarily for material, but for things you need to access for the workshop. We have several places where you can find information about the workshop. First is our website at CompuCell3D.org/workshop26. All relevant information about the workshop will be posted there. We try to keep that as up-to-date as possible. Information about the exact schedule of content for both weeks of the workshop will be up on the website soon. You will be receiving fairly regular update emails from me over the next several weeks in preparation for the workshop. So the emails will have all of the links you need and all of the latest information that is necessary to know about the workshop. But in case you can't find one of those, the information will be available on the website as well. If you do visit the website for something that you feel is important to know about the workshop and it's not there, please do let me know as soon as possible, and we'll make sure that that gets addressed. So you'll also need to download CompuCell, which you can do from the link at the top of the slide here, CompuCell3D.org/sourcebin. We'll have a second video on that will be linked in the description below about how to download and install CompuCell and how to test it once it is installed. You can reference our online help and manuals, as well as other help files. There's also a CompuCell3D Quick Start Guide. Now, it's important to note that this is based on CompuCell3D version 3.6. We're currently at version 4.8, soon to be 4.9. So the Quick Start Guide is a little bit out of date, but most of the core information is still the same. The biggest difference will be changes to the graphical user interface of the actual software. So it's still a good place to go to kind of pick up a few basic skills in CompuCell if you'd like to get a head start. Tellurium is the network modeling language that we'll be using in CompuCell during the workshop. And the quick reference guide is just a good thing to have access to during the workshop. It gives you a very quick rundown of basic CompuCell commands and cell properties and things like that. So again, our Slack channel will be used during the workshop mostly for kind of offline communication throughout the course of the workshop. Typically, the Slack tends to be a faster way to get help from people, both because the instructors that are monitoring the Slack and your peers will be able to respond to you. So we find that it's a good place for people to communicate outside of official workshop hours, particularly if you're planning on working on the hackathon. You can make a group for yourself in the Slack or yourself in your group, and you can use that to talk and share files and all of that kind of stuff. So we'll have different channels for different topics, and this will be a place for you to engage with your workshop mates as well, rather than just the instructional team. So please do join that as soon as possible, and we will begin using that a week or so before the workshop starts. A couple other quick reference guides. We have a subdirectory in the student materials folder, which is linked up here at tinyurl.com/cc3d2026 reference. This contains several reference guides, including our Python beginner's cheat sheet, our CompuCell quick reference guide, and a Tellurium quick reference guide. We find it useful if you have access to a printer to kind of print these out and have them in front of you during the workshop for quick and easy referencing, or at least have them saved as PDFs to your desktop so you always have them accessible in the same place. We also have some kind of suggested readings in that reference folder as well. So if you're interested in learning more about the background of CompuCell, about how to think about conceptualizing your model, we have a few useful resources in that folder if you'd like to take a quick skim through some of those. All right, so just a few last notes here to prepare for the class and the hackathon. Things to do to prepare for the course: make sure you can launch CompuCell3D on your local computer. And again, we have a video linked in the description about how to do that. There are a few demos that we recommend people run after installing CompuCell to test and make sure that everything works. In your local installation folder, they'll be in the demos subdirectory, and they're listed here: the steppable base mitosis, angiogenesis, and delta notch. These three simulations use a number of different CompuCell features, so if you can run all three of them, your installation is in good shape. If you have problems running any of them, then it's worth contacting us to make sure that something didn't go wrong with your installation. If you'd like to do any background reading to review any of the core concepts you don't feel comfortable with, you can obviously refer to the references linked below in the description or on the slides previously here. Just again to reiterate, you do not need to be an expert, but seeing some of these ideas before you have to work with them during the workshop will make things go a little bit easier. If you are planning to participate in the hackathon, think about a project you want to do or what kind of project you might want to join. We've also found it useful if you're planning on joining a group to provide some details about yourself, like your background, what experience you have in modeling, what experience you have in programming, these sorts of questions. And we actually have a teaming document which we'll be sending out a link to closer to the workshop for people to start sharing their ideas, and for people who are looking for a group to share that information for people looking to form groups to reference. And with that, we have reached the end of our orientation. Please feel free to submit any and all questions you may have to me, Hayden Fennell, this year's workshop organizer and coordinator, at hfennell@iu.edu, and do note that there's only one L in the email address. That's just how it was generated, but I am happy to answer any questions that anybody has or set up any appointments to discuss concerns or comments that people might have in advance of the workshop. So please don't hesitate to reach out if anything has been unclear or if you have any questions that were not answered by this orientation. So thank you for your time, and we will see you all at the end of July.